Hi students, this is Professor Fernetovich, and today I'm going to talk about cell membranes. This is mainly referring to the cell membrane on the outside of the cell, the outermost part of the cell. All cells have a cell membrane, but in eukaryotic cells, we also see the same type of membrane surrounding the nucleus. So keep that in mind as we go through today's lecture. The plasma membrane is mainly composed of a phospholipid bilayer. And a phospholipid, just to refresh your memory, has two fatty acid tails that are hydrophobic. They're nonpolar, so they repel water. And then there's a phosphate head, which is polar and hydrophilic. So the heads point out of the cell and into the cell, and the fatty acid tails are kind of wiggling towards each other. And I'll show you a couple pictures coming up. That's the main part of a plasma membrane. There are also proteins and carbohydrates throughout, as well as cholesterol. So the main function of the cell membrane is to contain the cell as a compartment on its own. The plasma membrane, both on the exterior of the cell and at the nucleus of a eukaryote, is selectively permeable, and that means that some things can cross it and some things can't. It's often described as a fluid mosaic model because those phospholipids, being lipids, have a degree of viscosity. They are kind of liquidy. Some are more liquidy than others. So that liquid fluidity of the plasma membrane can be analogous to a pool surface, like the surface of water. And there are beach balls floating on that surface. Those would be proteins. So proteins are all throughout the cell. They help with communication, and they help with movement across the membrane. The fluidity is also influenced by temperature. The higher the temperatures, the more fluid that membrane is. So warmer cells have a more fluid membrane. Think of like butter melting. When it gets cooler, the fluidity of the membrane decreases. And the fluidity is also influenced by membrane composition, whether we have saturated or unsaturated fatty acids in the phospholipid tails. That can affect it. Saturated fats are more solid, and unsaturated fats are more fluid. And those unsaturated fats can't pack as close together. Cholesterol can also influence the fluidity of the membrane by getting in between those fatty acid tails and keeping the cell from getting too fluid when it's warm and too rigid when it's cold. So here's a little picture of a plasma membrane. You can see the phospholipid bilayers, kind of like the brown sandwich here with the little tails sticking towards each other. So that gives a layer of hydrophobic material, and that helps to regulate the amount of water inside the cell and versus outside the cell. We also have the proteins. They can go all the way through, integral proteins, or they can be peripheral proteins just on one side or the other. And then glycoproteins have a sugar attached to the protein. Glyco refers to sugar, and that helps in cell-cell recognition. I mentioned cholesterol helping regulate the fluidity of the membrane, and this is very important. A lot of people think cholesterol is just all bad, but on the cell membrane, it's incredibly important. As the temperature increases, like I said, that fluidity increases, and the cell can get too loose, if you think of it that way. Cholesterol can prevent that from happening. And as temperature decreases, the fluidity decreases as well, but cholesterol can mitigate that and keep the cell from becoming too rigid and stiff. So membrane proteins make up the mosaic part of the fluid mosaic model, like I was talking about before. They're the beach balls floating on the surface of the pool. Those peripheral proteins are on the surface. We can see some here, as opposed to an integral protein going all the way through. Some integral proteins are solid, like this one, and some are hollow, like this one. And this is a channel protein that can allow small molecules to go across the membrane. Integral proteins can also help in transporting material through the membrane, and so can the peripheral proteins. They can act as shuttles. So those membrane proteins are very important for the cell for transport. They can be a site of enzymatic activity where molecular reactions are happening. They can help in communication, and they can aid in cell-cell recognition, particularly when they function as a glycoprotein. Now, crossing the cell membrane is different for different types of molecules. So let's talk about that. So a cell needs to get rid of waste, and it needs to take in certain substances. So how does that happen when we have this phospholipid bilayer and all these proteins? Well, hydrophobic molecules, those are nonpolar covalently bonded molecules, fatty things, they can dissolve easily in the lipid bilayer. So they can get in between the phosphate heads and travel through that bilayer. Certainly that's very easy for small molecules. Transport proteins can help assist other molecules, especially hydrophilic molecules. Channel proteins can act as a hydrophilic tunnel through the membrane, letting little tiny things go through. There are aquaporins, which are a great example of channel proteins that just let water through the membrane. There are carrier proteins as well that take up molecules and change shape to transport those hydrophilic molecules across the membrane. There's a picture here. So carrier proteins often require energy to do their function. So they will sometimes need what's called ATP, adenosine triphosphate, which is the energy currency for the cell, to do that, to move those molecules back and forth. Channel proteins and what we just saw previously with the small molecules just diffusing through the membrane don't take any energy, and that's called passive transport. Diffusion and osmosis are examples of this. Spontaneous, meaning no added energy is needed. This follows a concentration gradient from high to low, and we end up with even concentrations on both sides of the cell membrane. So passive transport diffusion, this occurs not just across the membrane of a cell, but whenever we have a concentration of particles put in a larger space. So if you're making lemonade, you might put in some lemonade powder and water, and we have that concentrated lemonade, and that will eventually diffuse throughout all the water and be evenly distributed, hopefully, and you don't have the sugar at the bottom. It happens in gases, and it even happens in semi-solids diffusion. So just keep in mind that it does not require any ATP or energy to occur. Osmosis is a special type of passive transport. It's like diffusion, but just for water. The sodium-potassium pump is an example of active transport where energy is required to move a substance across the membrane. We saw the picture of the protein opening for a molecule and then changing shape and releasing it on the other side. Well, that's what's going on here, but there's a little bit more to it. With the sodium-potassium pump, this regulates sodium and potassium ions inside the cell. And it's important because it causes an electrochemical gradient across the cell membrane, which can help in communication between cells, particularly nerve cells. So you'll see this if you take anatomy and physiology when you study neurons. This is a simple diagram showing the sodium-potassium pump. What we've got here is the phospholipid bilayer and a big protein. So it's open towards the inside of the cell in this first picture, and there are 3 circle-type spots there for 3 sodium ions. They're all +1 ions, and so they will attach into that protein, and ATP energy will be used to change the shape. The sodium will then get released out of the cell, and potassium will come in. So sodium's out, potassium comes in, and then we have 2 pluses inside the cell. So this creates a negative electrochemical gradient inside the cell. So the cytoplasm becomes negative compared to the outside of the cell. All cells have a voltage across the membrane, and it's those ions that create that gradient. So ions are fairly small molecules. Let's look at larger molecules. How are they transported across the membrane? Well, exocytosis is the process of releasing molecules out of the cell. So exo, like exit, cytosis for cell. Vesicles from the Golgi membranes fuse to the plasma membrane on the outside of the cell and dump out the contents. So here's a little vesicle from the Golgi. You can see it's wrapped up in a phospholipid bilayer, just like what makes up the plasma membrane, fuses with the membrane and releases maybe waste products or hormones outside of the cell. The opposite of that is endocytosis, where molecules, large molecules, come up to the plasma membrane outside the cell. A new vesicle forms at the membrane and the cell takes in the molecules. Endocytosis can take some different forms. There's pinocytosis, which describes when a cell gulps up fluid and important molecules, and phagocytosis, which means cell eating, where the cell engulfs a food molecule with pseudopodia. And you'll see some of these in lab when you look at amoeba. They extend these arm-like structures out of the cell, wrap around food particles, and bring those into the cell. There's also a special case called receptor-mediated endocytosis, and this is my favorite one. There are receptor sites in these little pockets of the cell membrane. So here's the cell membrane, here's inside the cell in yellow, outside is blue. And the little receptors will bond to specific molecules, form a concentrated vesicle, and bring that into the cell. So that's when a cell needs a specific thing and brings it in. So I hope this is helpful describing the membranes. I do want you to focus on diffusion and osmosis and look at the tonicity that's described in your textbook and in some of the exercises. Also, the sodium-potassium pump will be extremely important for those students going on to anatomy and physiology, so take a look at that as well. And as always, feel free to email me if you have any questions.